Good morning, everybody. Just want to begin by thanking the organizers for giving me the opportunity to attend this workshop. Yeah, it's been a wonderful education, learning from all the experts in the field as our laboratory tries to enter the computational realm with the rest of you. I'm going to begin by introducing the biology our laboratory is interested in. That's been largely led by Marie Porchualo, a student, and supported by various lab members, and then the computational analysis we are performing to try to really get at this question of how does anatomy instruct tumor cell states. So the area we focus on is melanoma. As many of you may know, melanoma arises following the malignant transformation of melanocytes. And melanocytes are very exciting cells where they exist across your body. You have melanocytes in your eye controlling eye color. You have melanocytes in your mouth, your heart. And depending on where anatomically that melanocyte exists, if it becomes transformed, it dictates the type of melanoma you'll get. So there are a variety of different types of melanoma that exist. Most of the studies in the field have been on cutaneous melanoma, those associated with UV exposure. That's why you need to wear sunblock. And this is primarily superficial spreading melanoma. Our laboratory focuses on acral lentiginous melanoma. This is on sun-protected sites, on your hands, your fingernail beds, and the bottoms of your feet. This is not associated with UV. Been barely studied in the field, and because of that, there is not a single FDA-approved treatment specifically for these patients. And so it's not a surprise that these patients suffer worse 5-year overall survival rates compared to cutaneous melanoma, and they suffer elevated rates of metastasis. Now, what the field and many have boiled down this property of metastasis being elevated in acral is just as a consequence that you're not really looking on the bottom of your foot or your hands. Maybe people miss it, and that's why people present at a more advanced stage. So this is what's been published as a likely explanation, just a thicker tumor, more advanced stage of presentation. However, recent groups have found that if you normalize stage of presentation tumor thickness, you still find that these acral melanomas are much more metastatic compared to cutaneous melanoma, which tells us that something biological is likely at hand. So the way our laboratory focuses upon this problem is through the lens of nature versus nurture. Is it due to the unique nature of acral, defined as their tumor cell intrinsic differences that are responsible for that elevated metastatic capacity, or is it due to the element of nurture, defined as the unique tumor cell extrinsic differences in acral versus cutaneous melanoma? Indeed, when we look at nature, acral melanomas are unique in that they have significantly greater frequency of structural rearrangements. They have a unique genetic evolution compared to cutaneous melanoma, unique drivers, as well as a transcriptionally defined identity that can stratify a acral versus cutaneous. When you look at the element of nurture, it's also quite distinct. Acral melanomas arise on the hands, the nail beds, and most frequently on the bottom of the foot. These environments have unique biomechanical properties, immune landscapes, and microenvironment composition. So our laboratory, and in the next few slides, I'll show you how we're exploring this phenomena, this phenotype that we're interested in, metastasis, through the lens of nature versus nurture in acral melanoma. So to begin, we acquired some beautiful clinical samples. We have a cohort of 17 plantar acral melanomas. So plantar is the bottom of your foot. And we have 540 cutaneous melanomas. These cohort had collective text imaging reports that allowed the application of a natural language program that could identify specific metastatic sites in patients over time. When we look at the genetic properties of these plantar acral melanomas versus the cutaneous melanoma patients, we find that the things that have been reported in the literature hold true. These acral melanomas have lower tumor mutational burden. There's less UV irradiation. They have less UV signatures. And when we look at the metastatic property, we find that these plantar acral melanomas have significantly greater metastatic rates compared to cutaneous. On the y-axis is mean cumulative metastatic sites. Now, from this cohort, we can't really say nothing about nature versus nurture, given how different the nature and nurture is between these plantar acral melanomas and the cutaneous melanomas. So then we asked, is it possible to try to find a clinical cohort that was much more similar in nature to allow us to determine whether the element of nurture might be driving this metastatic capacity? Luckily, we were able to identify 13 dorsal acral melanomas. So these are acral melanomas on the top of the foot, to compare with the acral melanomas on the bottom of the foot. Top of the foot has very distinct biomechanical features compared to the bottom of the foot. When we look at their genetic features, we find that these two different cohorts are very similar in their nature. So it provided us an opportunity to see whether the property of nurture in the bottom of that foot, the plantar skin, is driving that metastatic capacity compared to the top of the foot. And indeed, when we look at the metastatic capacity, we find compared to dorsal acral melanomas that don't have much metastatic capacity compared to cutaneous, we find that those plantar acral melanomas, again, display an increased distant rate of metastasis, suggesting that this element of nurture might be partly responsible for this elevated metastatic capacity in plantar acral melanoma. So these are certainly interesting data, but we then asked, is there a way to potentially better experimentally test this variable of nature versus nurture in a controlled way using mouse models? And so over the last 5 years, our laboratory has optimized a series of orthotopic mouse models where we keep constant the tumor cell. We either use murine melanoma cells or human melanoma cells. We keep that constant, and the only thing we're changing is the tissue we're implanting these tumor cells in, either the plantar skin to model acral melanoma or in the non-plantar skin of the flank to model cutaneous melanoma. So same tumors, different tissue, and we're asking the question, does the tissue instruct the metastatic behavior we're seeing clinically? In this first series of experiments, we used the murine melanoma cell line YUM 4.1. We functionalized them with an M-cherry luciferase tag so that we could monitor metastasis using intravital imaging. We can confirm that the tumors grow inside of the foot of mouse. This was not something that was clear, not in the literature. We see that the tumors grow much more slowly when implanted in the plantar skin, shown here in peach, compared to the non-plantar skin. However, the metastatic capacity was significantly increased. These tumors implanted in the plantar skin of the foot demonstrated significant elevation in their ability to metastasize to the liver versus non-plantar skin. In the back?
Sorry, did you quantify the mechanical property differences between the two?
Yeah, and a couple slides will have actual functional data on that. So we repeated this experiment now. Instead of using an implantation model, we used a genetically engineered mouse model approach, which was just recently described a little bit by Russell. Here, this genetically engineered mouse model allows you to transform melanocytes wherever you deliver this Cree virus. Triggers the formation of these mutations. This has been developed by our collaborator, Sherry Holman at the Huntsman. Sherry has published quite a few papers inducing melanomas at cutaneous sites. And so we asked her, could you try to induce the transformation of these melanocytes in the foot pad so that we can compare side by side the metastatic capacity? So that's exactly what was performed. And again, we find here that when we induce the transformation of plantar melanomas in a mouse, again, they demonstrate elevated metastatic capacity compared to those non-plantar melanomas. And then, of course, we really care about human acral melanoma. So we repeated the exercise now using human acral melanoma cell lines. Again, we see that implantation in the plantar skin has a slower tumor growth rate, but again, an elevated metastatic capacity in this model to the lungs. It's not always the same organ. We see some genotype-specific phenomena. All in all, we believe that, at least with these studies, the element of nurture appears to be significantly contributing to that elevated metastatic capacity we see in patients. We find that orthotopic implantation or induction of de novo melanomas increases that metastatic potential. And the plantar skin appears, again, to be nurturing this. And so we were curious what exactly changed.
Sorry, I'm outside the field. Can you explain the difference between metastatic potential and metastatic capacity and how they're
synonymous?
And what does it mean percentage? Like what's the observable of capacity?
The observable is we're just monitoring of the total mice that were either implanted in the foot versus flank or the tumors were induced, the frequency of mice that then demonstrated metastatic disease in their organs. Yeah, it's a synonymous. I should be more consistent when I'm saying it. So we were curious, obviously, what exactly changed. Why are these tumor cells in the foot pad, the plantar skin, becoming more metastatic? So this is where we began being interested in cell state shifts and how the plantar skin microenvironment is shaping the cell state. To begin the study of the cell state consequences, we first looked back at this study published 5 years ago, where they defined a transcriptional acral melanoma identity. The way they developed this identity was by first looking at healthy melanocytes in the plantar skin. Another word for plantar, volar. So these are these volar melanocytes, and then looking at healthy cutaneous melanocytes, they call CMEL. And so what they did was they created a ratio of the genes upregulated in volar acral melanocytes versus those genes upregulated in cutaneous melanocytes. And what they found was that you could stratify a acral melanoma from a cutaneous primary melanoma. And this makes poetic sense because a acral melanoma would arise from an acral melanocyte being transformed. So many in the field of acral melanoma believe the reasons biologically acral melanomas are different from cutaneous melanomas is because the melanocytes they come from, the cell of origin, are so distinct biologically, and those differences are maintained inherited. So our question was, is this identity hardwired? Is this part of the nature of acral versus cutaneous, or is this another situation that may be nurtured by the plantar skin microenvironment? So taking advantage of our non-plantar and plantar approaches, only changing the anatomic site, same tissue, same tumor.
What is the melanocyte density difference between, like, are they very, very rare within the plantar skin?
They're on the bottom. They're not as dense. Their function is unclear at the moment because you're not protecting against sun. Some believe maybe thermal regulation in unpublished studies, but they're there and they're there.
The fact that you saw those differences in there was that study that you studied initially where it was like the top of the foot versus the bottom of the foot. I mean, we walk on the bottom of our feet, not on the top of our feet.
Yeah.
So is there like some kind of, environmental difference that has to do with the friction? Or,
Yeah.
like it's like UV kind of in some way.
In terms of the etiology, that's what many believe, that it must be something to do with maybe injury or all the compression, stretching that occurs. Stiffness is elevated, the collagen density is elevated. There's a lot of biomechanical differences. I'll have some slides showing one specific biomechanical property we then show functionally plays a role here, but these are all open questions we're very interested in. Just not been experimentally demonstrated before. Exactly. All right. So looking at the acral melanoma transcriptional state, if what the field believes is true, we should not see any shift in the acral identity. If the field is correct that it's hardwired, it shouldn't matter if we stick it in the foot, the flank, the ear. So what we find is that when we look at our murine melanoma models and our human melanoma models, when we implant them in the plantar skin, we see a significant enrichment of that human acral melanoma transcriptionally defined identity. So they appear more cutaneous-like when they're in the non-plantar skin of the flank, and they appear more acral-like when they're in the plantar skin of the foot. We then looked to try to, as much as possible, validate these findings with different independent data sets, different laboratories. So next we collaborated with Casey Coots. She runs a rare melanoma translational core at Colorado University. So Casey's goal is she's trying to make PDX models, patient-derived xenograft models. She's trying to make tons of tumor tissue. So she implants cutaneous melanomas and acral melanomas all in the non-plantar skin of the flank, because her goal is just trying to expand to share. So she's created many bulk RNA-seq data sets. When we look at the cutaneous and acral melanomas all in the non-plantar skin of the flank, we don't see any stratification of that acral melanoma identity. When we look at publicly available bulk RNA-seq data sets of plastic cultured cutaneous melanoma, acral melanoma cells, we do not see any difference in that acral melanoma identity.
So the experiments in the upper right, those are nude mice?
Those are all nude mice, yeah.
That's one, I guess, big difference.
One possible, but in our studies, the murine are in immunocompetent, the human are in NSG, so it doesn't seem to be an immune-related thing. And then we went back to the clinical data. We validated in an independent clinical cohort what was published 5 years ago. We again could see that you could stratify primary acral versus primary cutaneous, but when we look now at the metastatic acral versus metastatic cutaneous, you lose that stratification, suggesting all in all that the plantar skin microenvironment is what allows the enrichment of that identity. So this points to an identity crisis of sorts. What people believe is defining of acral versus cutaneous does not appear to be hardwired. It's something that is plastic induced, and it is the microenvironment that allows a melanoma cell to look acral-like or cutaneous-like. And so that is the hypothesis our laboratory is exploring, how the identity is being nurtured by the plantar skin microenvironment. So as you can imagine, and that was what Sam alluded to, what in the world in the plantar skin microenvironment is potentially shaping this? There's so many differences. The difference that we focus on for some of these studies initially is that the stiffness in the plantar skin microenvironment is significantly elevated compared to non-plantar skin. And so when we culture acral melanoma cells and other types of melanoma on the stiffnesses that are observed in plantar skin versus non-plantar skin, we again see that culture on a stiff matrix increases, enriches that human acral melanoma identity. Also, when we look at other different gene sets that are enriched, we find things that have been published before. We see that there's increases in adhesion signaling, Rho GDPases, the actin myosin contraction, YAP activity, as well as programs associated with melanoma metastasis and invasion. This is also functional because when we prime these melanoma cells on soft and stiff, we also see that they are functionally more invasive when then tested in Boyding chamber assays. All to say, it appears that stiffness appears to be one of the key regulators in the plantar skin microenvironment of that human acral melanoma cell identity. So up to this point, we have shown with multiple independent in vivo models that the plantar microenvironment is changing tumor behavior. It's making them more metastatic. It's making them more acral identity-like. Obviously, behavior is a consequence of cell state. And so we are now interested in trying to understand how the cell state is changed by plantar microenvironments, by performing many omics and trying to make sense of the data. So far what we've done is we've collected the tumor cells from the non-plantar skin versus plantar skin, and we've performed single-cell RNA-seq and spatial transcriptomics with heavy collaboration from Johan Gudjonsson at the University of Michigan. And then we've also done multiplex immunofluorescent microscopy with the Peter Sorger lab at Harvard. For the sake of time, I'm going to focus on what is occurring specifically in the tumor cells in that single-cell RNA-seq data set. And so this is being analyzed by our staff bioinformatician, Marjorie. So here is the total tumor microenvironment composition. She is filtering out just the tumor cells, and you see already that the plantar tumor cells are separating quite nicely from the non-plantar implanted tumor cells. Here is just showing the different mouse tumor replicates, showing pretty tight clustering of the non-plantar versus plantar. So obviously we're interested in what transcriptional programs might explain these biological differences we're seeing phenotypically. So CNMF was run by heavy advice from Yogesh and members of his lab, Leon and Nate. So what Marjorie did is first she tried to determine the number of programs, the K value that had maximum stability for the non-plantar tumors as well as the plantar tumors. So the choice was, so I'm presenting this in a way also for as much advice as you guys can provide, because this is very new to us. This is what we've tried so far. So the K value that was selected for the non-plantar was 5 because of max stability. In the plantar skin, 7. What we then did was, so here is just showing then those programs, the 5 programs in the non-plantar skin and the 7 programs in the plantar skin. So then here are the different marker genes associated with those different programs, 2 extra in the plantar context. And there is congruence in the same marker genes are appearing in the same, in programs across 2 different sites. And so at this moment, then we're trying to either, well, both things. We want to discover new programs potentially that have not been described. And then also option 2 is to see how we can map programs onto published melanoma data sets, melanoma states. All that's been done so far in the melanoma field has been really in cutaneous melanoma. We're interested to see, is there complete agreements or partial agreements or no agreement now that we're looking at acral melanoma in that plantar environment. And then also something that we're curious about is how can we best combine these 2 approaches, these 2 goals. So what we've done so far is the option 2 benchmarking approach. We've looked at previously published melanoma cell states coming from the Christopher Marine Laboratory. There's been more than 11 different cell states documented so far. Here we are using as reference either the plantar K that was determined when deciding what was the most stable, and then for the plantar data set, and then the non-plantar K that was determined for then the non-plantar data set. And so what we see when using site-specific references is that we do see certain cell states that have been previously reported to be enriched in our plantar skin implanted tumors versus the non-plantar flank. A lot of these cell states have been associated with increased aggressiveness, increased metastatic capacity, reduced sensitivity to immunotherapy. So these are cell states that make sense with the clinical data, but this is how we see it when using site-specific references. Through the advice from some in the audience, we also tried using a shared reference. So putting all the plantar and non-plantar tumor cells together, identifying what might be the maximum stability K-score when performing CNMF. And when we perform it in this manner, we lose any significant differences across the programs in the plantar versus non-plantar site. So this is where we are at the moment. Many of these analyses are very new to this world. And so the advice that we're looking for, the questions that we pose is in terms of the biology, how do we best determine whether the plantar tumors are adopting new cell states or simply this is an enrichment, a selection of pre-existing cell states? Some of the way we're thinking of answering this is with Yogesh performing those lineage barcode approaches. In terms of a computational question, should transcriptional programs be learned separately or for each tissue or jointly across the tissues? Annotation question, should the cell states be assigned discretely or represented as continuous mixtures, continuous. And then the validation question, what kind of orthogonal evidence would convince that these differences are biological rather than analytical? So those are the questions that we're hoping to get advice, and have already gotten a good deal of advice from the community. This is the team that's done all the work, the A team. We have a ton of great collaborators across the globe. And yeah, looking to try to get the advice from the experts in the room, how to best proceed. So with that, I think I was the first one to end pretty quick.
Great work. How much of this do you think is mechanical versus actual cell state change driven in terms of functional outcomes? Have you tried to induce the cell state in the absence of placement in the plantar foot and see if it drives greater metastatic potential, or is it that they're confounded with some common other stimulation?
Yeah. I mean, with the stiffness, and we have some other studies looking at compression, stretching, we see that the biomechanical acral properties, the plantar properties, appear to induce that identity. Whether the genes that constitute the identity are functional is unclear. We also see what appears to be a stiffness-induced transcription factor, that when we silence or overexpress that transcription factor, we can phenocopy outside of the plantar skin some of those same behaviors. So that does appear to be potentially in that manner, but yeah, still more studies.
And is this contrary to some of the results from Ashi's lab that there was, greater coherence of the collagen matrix? Does that correlate with stiffness and leading to changes in metastatic potential?
From what I've learned when talking to experts in the collagen world is that the collagen abundance is not one-to-one with stiffness.
Okay.
And sometimes those get conflated. The orientation of the collagen, the bundling, sometimes you could have less collagen abundance, but the orientation is different and it could still be stiffer.
Okay.
But all in all, there's nothing that's in disagreement with what she's been seeing. And in her laboratory is another cutaneous melanoma laboratory where they look at age. They see a similar pattern where in aging mice, when the tumor cells grow slower at the flank site, they're more metastatic. So it appears to be a common phenotype switch where in melanoma, the proliferation may be exchanged for invasive capacity.
Is it really clear that the tumor cells themselves are growing more slowly as opposed to the tumor being broken up a lot, for example, just because of the mechanical?
Yeah.
And then likewise, cutaneously, maybe it's not that the cells are proliferating more slowly, but that the tumors are smaller if the cells are metastasizing.
Yeah. Those are very interesting questions that we're trying to get at. Is it smaller because it's just more edited? Is it more senescent or more cell death occurring? Those are unclear at the moment, but those are things we're looking into. It's a good question.
Before Christian on your screen, will you just show the box plots again 1 second? I'm just thinking.
Here?
Yeah. Oh, there we go. I'm just thinking. So those are actually right. You have weights, right? So for each mouse, you have a distribution of weights. So I don't know how CNMF. I don't know. You'd have to maybe do an empirical null or something. But couldn't you look and see if, how these fractions differ is really determined based on what you call a cell that has that program, right? Like what, but you could just look for the distribution and say, is there a shift in the 4 mice that have if I look at program whatever one or whatever the green thing is, that really does look different. Maybe the 4 mice have a higher distribution and there's, it's just that you're picking a threshold that's slightly weird. And if you looked at distribution of them, you'd actually be able to see something about the mean shifting or something like that. I don't know. Yeah. I don't know exactly I don't know if you could do a t-test based on the constraints here, but something like that.
That's a good point. That's something we'll make sure Marjorie does.
If you ask for a picky detail, that's the thing. Now Peter and I are going to have a fight outside.
Yeah, I enjoyed the fight. But something to appreciate too is, for example, the neurocrest, there's no difference in the program frequency between the plantar and the non-plantar, but the different gene sets that are associated. There could still be extremely distinct biology.
Right.
A different flavor of neurocrest that's
totally. That would be another thing that would be really cool to look at.
All right. Great. Thank you very much, Vito. Let's definitely ask Vito more questions over lunch.